Bismillahirrahmanirrahim. This is part two of our lecture series on the nucleus. We discussed certain aspects of the nucleus, just like we discussed about its discovery, the space that the nucleus occupies within the cell. It's a tiny space where a large fragment of the DNA, about 5.9 feet long DNA, is enclosed within this tiny space. And then we discussed about different structural elements of the nucleus. We discussed nuclear membrane in detail in the last lecture. And then we were discussing the nuclear pore complex. And other structures will be discussed in this sequence as given here. This is the nuclear envelope that we discussed, that it's a double phospholipid bilayer. There are two layers of phospholipids. In the case of plasma membrane, you can find only one layer, but here are two layers, and there is space in between, which is called the perinuclear space. And the rest of the properties of this membrane are essentially the same because it's a phospholipid bilayer. So you can just imagine about its role, that how lipid-based substances would get through this particular structure, but not charged substances or very large molecules or polar molecules would not be able to cross this particular membrane, and that would require special structure in the form of different kind of proteins which would help substances to get through them. And these are the nuclear pores. These are different kind of proteins which have come together to make pores in this envelope, and these are helping with substances to get through, to get inside the nucleus or to get out of the nucleus. And this double membrane, the outer membrane is an extension of the rough endoplasmic reticulum. All the membranes inside the cell are essentially phospholipid membranes, and uh, it is also the case with that of the endoplasmic reticulum. So the rough endoplasmic reticulum extends to make this outer layer or outer membrane of the nucleus. So this is the outer membrane, and then there is the inner membrane, which is dark pink in color, and it is supported by this lamina, which is a matrix-like structure made up of different proteins in order to make to support the structure of the nucleus. So this endoplasmic reticulum and this outer membrane of the nucleus, which are essentially the same thing, are studied with ribosomes. And what are ribosomes? These are protein making machineries. And these this particular structure, the endoplasmic reticulum, is important because proteins are synthesized here, and then these proteins are transported to different parts of the cell for different functions. So we discussed about the nuclear membrane. We also discussed about the disintegration of the envelope of the membrane, which happens during the process of mitosis when the cell is going to divide. And we discussed about the CDKs, or the role of the cyclin-dependent kinases, which phosphorylate these proteins. And after phosphorylation, some of the complex disintegrate from the entire complex, thus crumbling down the entire complex into pieces. And these species are sometimes stable, and they can rejoin and remake the same structure when the cell division is complete. And the CDKs not only are disintegrating the membrane, uh, but they are also disintegrating the laminin, or the laminin proteins, which are making the support structure on the inner side of the inner uh, nuclear membrane. So both the structure are disintegrated and integrated again when needed, when the cell has to divide, when the cell has almost completed the rest of the division process. Then we were discussing the nuclear pore complexes. I told you that passage through the phospholipid membrane is not easy for many substances, just like charged substances cannot get through the phospholipid membrane. Similarly, substances that are very large in size cannot get through it. Water cannot get through this particular plasma, this particular phospholipid bilayer membrane, which is double phospholipid bilayer membrane around the nucleus, providing it extra protection, and a continuum with the endoplasmic reticulum, which is needed for the transportation of different proteins on urgent basis, because lots of proteins are required inside uh, the, the nucleus for different functions, like different transcription factors. That machinery requires different kind of proteins on regular basis on a massive scale. So that structure is continuous with the nucleus. So that's problem is solved by this particular mechanism. Now, the transportation is of two types: active transportation in and out of the nucleus, or passive transportation. So active transportation in the case of large molecules or charged molecules, etc., which is called active because it is an energy dependent process. Energy will be required for this particular process to push something out of the nucleus, or to pull something into the nucleus. In and out transportation would require energy, so you have to fuel this particular transportation. Here the passive transportation also occur in the case of ions or small molecules, and it happens as it would happen. It doesn't require any kind of energy. And this nuclear pore complex, which is composed of different kind of porins or proteins, nuclear porins they are called, these complexes span from the lamina, which is the support structure on the inner side of the membrane, to the outer membrane, or the outer side of the the nuclear membrane, as I showed you, from this particular blue lining to this particular. So the pores are here, and the pores are through and through. You can see that the pore is extending from the outer membrane to the lamina. So this pore, actually the pore proteins, they are connecting this entire structure, or reinforcing this entire structure instead. Um, if you look at the number of pores in a mammalian cells, there may be 3,000 to 4,000 pores in a mammalian cell. And these are tiny holes. These pores are tiny holes made up of protein molecules which come together. And the diameter of a typical pore is about 100 nanometer, but the gap for molecule is only 39 nanometer because of multiple proteins. When the nucleoporins are these new proteins, they come together to make a hole-like structure. That hole-like structure is the diameter on, if you look at the diameter from the upper side, it's about 100 nanometer, but if you look at the space which is provided for the entry or exit of molecule, that is only 39 nanometer. So these proteins, they are called NUPs, and there are a number of NUPs in different species, like maybe more than 30 in yeast in different species. And vertebrates, there are about 30 identified NUPs, or nucleoporids. So there are lots of proteins which are making up, these pore complexes. We call them complexes because these proteins would come together and make a structure like a complex. That's why we call it a complex rather than just pore. So these porins, they have a larger diameter on the upper side, but when these proteins, they come in different forms, and when they attach to each other, on the inner side, the pore size is only 39 nanometer, which doesn't allow many substances to get through it. So many of the substances which would get through the pores would need some kind of help from other sources, or very big substances may not enter into this. It's the case with many different types of viruses as well. That why some viruses, they can't enter into the nucleus, while some viruses they can get into the nucleus, or their DNA just get into the nucleus, while some can't do that particular thing. We would discuss about it in later sites. So these proteins are also called karyophorins, and we divide karyophorins in two categories: importance and exportins. Importance are proteins which are required for importing substances into the nucleus, and there are two forms, alpha and beta forms of the importance. Um, and exportins are proteins which are required for exporting, substances from the nucleus into, the cytoplasm. So parents is a family of transport factors for proteins, ribosomal subunits which are there, and some DNA. So all these substances need the help of different kinds of carrier of the proteins, importance and exportants, to get into the nucleus and come out of the nucleus. And excuse me, some carrier are some of these important and exportant proteins. They are, mobile in nature. Usually people think that, these proteins would be static in nature and they would be fixed to certain positions, but there is plenty of evidence suggesting that many of these caryophorins or the nucleoporins, they are mobile in nature. You can find these proteins in different parts of this tunnel or this spore that they make on the cytoplasmic side. Sometimes they are on the nuclear sides. Sometimes they are in the inter membrane space. So you can find them at different places, and they do different jobs at different spots. Most most of them directly engage target proteins, while some use adapter proteins. So some of them would just go there and directly grab a cargo and get it into the nucleus, and some need the help of some adapter proteins. But in all these cases, you should remember that nuclear localization signal is required for the entry of any protein molecule or any molecule that has to get through this pore complex, because without nuclear localization signal, none of the substances can get into the nucleus. That is the pass or the key to the entry of the nucleus. So now this is a consensus model of the nuclear pore complex. If you look at it, these are different types of proteins which have come together. This is the outer membrane and this is the inner membrane. It's just representation of the membrane. And this blue, golden, and this pink color, these are different proteins which are coming together. There are different cytoplasmic filaments of this particular structure which may help with the cargo. There are 8 different cytoplasmic filaments, and there are 8 different filaments on the inner side, on the nuclear side, are beyond the inner membrane. And so these different proteins which are coming together are making this structure. Are there? You can see these tunnels, um, which also, uh, you know, uh, they have different conformations. That's why when they come together, they would make this particular structure, or a structure just like this one, making a nuclear basket here where it can hold something. There's a distal ring, intranuclear filaments, just like the cytoplasmic filaments. There are also filaments on the inner side, which are known as, the intranuclear filament. So there is a cytoplasmic ring here, and there is a nuclear ring here. So a ring on the upper side and the lower side, and there is a nuclear basket here and a distal ring also here. So all these rings and baskets are made up of proteins of different natures. As I told you, 30 different kinds of proteins are making this particular structure, and they are known as NUPs. So NUP 78 or NUP 80 or NUP 153. There are different types of proteins, and they have different functions as well. Uh, there are lots of diseases associated with the nuclear pore complex, like several autoimmune diseases are caused by autoantibodies recognizing nuclear antigens. For instance, systemic lupus erythematosus or rheumatoid arthritis and primary biliary cirrhosis or PBC have been associated with proteins of the nuclear pore complex. Antibodies, which are generated by your body, sometimes they would target antigens or proteins of the same body, your body. So in that case, that kind of situation is known as autoimmunity. So it means your own immune bodies, the antibodies produced by your own immune cells, are recognizing your own body or cellular proteins as antigens, and they attack them and destroy them. So similar is the case with these nuclear pore complex proteins, that these react with these antibodies or autoantibodies which have been associated, autoantibodies which have been analyzed to react with these particular nuclear pore complex proteins. So different pore complex proteins have been associated um, with autoimmunity in these conditions like rheumatoid arthritis and cirrhosis. Different kind of cancers, leukemias and other cancers have been associated with the nuclear pore complex proteins like NUP 188. It's a nucleoporin 188 has been associated with malignancies, different kind of malignancies. In different kind of malignancies, when the tumor starts spreading to different tissues, we call it malignancy, malignant tumor. So this particular uh, nuclear pore complex protein has been associated with many malignancies. It is overexpressed in many malignancies, suggesting that it has got certain role in this particular malignancy. Similarly, the nuclear pore complex is involved in the entry of viral nucleic acids into the nucleus. Uh, as I told you, the diameter of the nuclear pore complex is 100 nanometer. But here you can say the space that is provided there for the entry of molecule is only 39 nanometer. NPC, this is not the diameter, sorry. This is the space allowed for the molecule. The entire diameter is 100 nanometer, but you can just say it's 39 nanometer space for the entry of molecules or exit of molecules from the nucleus. So certain viruses, they are smaller than 39 nanometer. For instance, the parvovirus MVM, it's about 25 nanometer only, or hepatitis B virus, which is 36 nanometer. So these viruses, it means that these viruses can pass through the nuclear pore complex in their intact form. So you can just look at the entry of the parvovirus here. The parvovirus, for instance, it reaches there at the nuclear pore complex. It's very small, 25 nanometer, and the hole here is wide open, so the parvovirus would just, uh, enter into this particular structure with the help of these importance and exportants. These blue are important beta, and red are important alpha. With the help of these proteins, the parvovirus would just enter into the nucleus in its intact form, and uncoating would take place over there. And in the case of hepatitis B virus, for instance, uh, again the particle is smaller, 36 nanometer. The space is 39 nanometer. So the the virus would be translocated into the nucleus along with the capsid. The capsid translocation and disassembly would take place in the nucleus. So they don't have to encode here at the nuclear pore complex. They would just get through them. So the particles which are synthesized there in the cytoplasm can get into the nucleus, while some of the viruses they have to encode on the outer side of the nuclear pore complex, just like the herpes simplex virus, which is comparatively larger in size, more than 39 nanometer in size. So these viruses their capsid cannot get into the nucleus. Important beta are involved in this particular process, but not alpha. Here, both alpha and beta importance are involved in the case of parvo and hepatitis B virus, but not in the case of herpes simplex virus here, where important beta only are involved in the process to just get hold of this particular virion or its capsid, and then uncoding takes place here, the DNA injection in translocation. So here it's a kind of injection that only the DNA of the virus is going inside the nucleus, but not the capsid. But here, in case of smaller viruses, the intact virus is getting inside and encoding inside. Similarly, some of in the case of certain other viruses, the capsid would be disintegrated right here at the nuclear pore complex, and then the nuclear material would get inside the cell. So that's why some of the viruses they um, can have access to the nucleus, while others cannot have access to the nucleus. So these nuclear pore complexes are not only involved in normal transportation of materials into and outside of the cell. These complexes, as they are made of diverse types of proteins, these are involved in different conditions, and also viral entry. That's why some viruses would get to the into the nucleus, while others will not. Then there are bodies inside the nucleus which are known as nucleoli or nucleolus singularly. They are one and two in or two in number. They are more in protein producing cells. Uh, they have a role in the ribosomes, making up the ribosomes, ribosomal assemblies. That's why they are vital to production of proteins. And they are densely packed. We don't call them organelles because they are not membrane bound, and they have tandem repeats of ribosomal DNA. And this ribosomal DNA is transcribed into ribosomal RNA by an enzyme which is known as RNA polymerase 1. And then when it is transcribed into ribosomal RNA, this ribosomal RNA is cleaved into ribosomal subunits. For instance, in the case of eukaryotes, this is cleaved into 5.8S, 18S, and 28S, sub units. The ribosomes, small and larger subunits, the proteins, of course, would be made inside the cytoplasm. They are transported into the nucleus, and inside the nucleolus, they join the repetitive segments of the ribosomal RNA, which is known as the functional assembly. So we call it a functional assembly of the ribosome that these units join together along with this ribosomal RNA. So this is a functional assembly, and this assembly would then leave the nucleus through these nuclear pores. And lots of ribosomes, functional ribosomes, may be needed by the cell. And according to estimates, 20 to 30,000 ribosomes are made per minute. That is the requirement of the cell because lots of different kinds of proteins are made inside the cells. And you know about the role of the ribosomes. We don't have to go into the details of ribosomes here. We would have a separate lectures on that. Ribosomes, just like mitochondria and other things, are also thought to belong to the RNA world, and ribosomes are thought to be one's self-replicating, you know, entities, or these ribosomes were, even before uh, the advent of the eukaryotic or prokaryotic cells were in existence, and they learned how to join different peptides together, how to make peptides, and they just got developed this association with the eukaryotic cell after entry into such kind of cells, but they were independent. They are thought to have existed as independent organisms, just like other organelles like mitochondria and other clusters, etc. So we would go into the details of the nucleolus and the ribosomes later, and their role in making proteins. These are protein-making machineries. How they make proteins, you would have gone through this particular thing in your molecular biology classes. There are some other nuclear bodies as well. For instance, the cadal bodies. These are coiled bodies, zero point 0.2 to 2 micrometer in size, and they are involved in the synthesis of small nuclear RNAs or snRNAs. The cadal body is the site of initial modification and assembly of several snRNPs are ribonuclear proteins, small nuclear ribonuclear proteins newly imported from the cytoplasm by the SMN complex. So this is SMN complex transport snRNPs into the cytoplasm, and its modifications take place in certain bodies inside the nucleus which are known as the cadal bodies, and they have a role in histone mRNA modification and splicing. The spliceosome, which is involved in cutting out introns from exons when transcription is taking place. So at that particular time, these spliceosomes are doing the job. So the spliceosome formation, these cadal bodies have a role in spliceosome formations. We call them coiled bodies because there is a protein which is known as coilin, and this coilin actually joins with this thing SMN complex, and then this complex again, it joins with these ribonuclear proteins to make the spliceosome. So, for instance, this is snRNA, small nuclear RNA, which is coming from the chromatin, and the small nuclear RNA in the cytoplasm is translated into SMN. And this SMN again, it enters into the nucleus, and inside the the cadal bodies, snRNP and SMN, they would join together, and then they are transferred. These, snRNPs join them, and then they travel to the speckle ICG, which is a place where uh, other, splicing machinery is assembled. So a functional spliceosome is made with the help of these cadal bodies, and that is needed for cutting out introns from exons when transcription is taking place. A protein formation ensues after that when mature mRNA is formed. In mature mRNA, there are no introns, and that actually, credit goes to the spliceosomes and the cadal bodies here. There are Gemini bodies and other, small bodies inside the nucleus. So these bodies would be discussed later in certain other lectures, but just you have to remember about the cadal bodies and spliceosome here. or the small nuclear proteins uh, in this lecture. How the nuclear transport is active or passive. In active transport, the transport is controlled by GTPases. There are GTPases controlled system there. The transport via importance and exportins is taking place. Important alpha and beta exportins are involved in the transportation in and out, and in this process, the GTP, uh, guanosine triphosphate, is converted into GDP with a free phosphate group and free energy. And that energy is used for importing substances or exporting substances or cargo from inside or outside the cell. Okay. So this is an energy dependent process, as I told you before, and rain is the key GDPAs, which is controlling, this particular process. Rain GDPAs is important here. For instance, there is a cargo; it has to go inside, so it would just attach with important alpha or beta. So important plus cargo would get into the nucleus. In here, it dissociates with the help of rain GDP. So the rain GDP would dissociate it, this particular cargo from the important. The important would be free and cargo would be inside. Okay. For instance, this is import. You can just look at this picture and you can guess that how the rain GDP actually is involved in this particular process, that rain GDP attaches to the cargo, and the cargo is then taken, uh, you know the cargo is taken inside. It's importing, so we are discussing import. The cargo in the rain GDP would just make a bond, and this cargo would get inside. The RAN GTP would act upon it, so the RAN GTP would attach with important, and the cargo would be released here. So you can find the cargo here. And the opposite to that, if something has to be taken out, then the opposite of this particular process would take place. And there are certain other factors involved in the transportation, but that is not the focus of this lecture. It's just about uh, to briefly tell you about the active GTPases based control system, and the other one was passive transportation. And you know about passive transportation very well, that how things move along a particular gradient. So that was this was about the nucleus. Further structures and other things would be discussed in later um, lectures. Thank you so much for watching.